Hey guys, so this lecture is going to be on biotechnology, and what we're going to do is we're going to examine several different techniques that are used and kind of fall under this biotechnology umbrella. We're going to go through them pretty quickly in this lecture, but then we will be revisiting things and talking about certain aspects of biotechnology in more detail during class time. The very first thing that we're going to talk about is recombinant DNA. So if you look at the word recombinant, it looks like to recombine, and that's essentially what you're doing. So you're taking DNA, or you're looking at DNA, which has genes from different sources, and they become linked together. So here you're looking at green mice. Mice aren't normally green. They also don't normally glow in the dark. And so what happened here is that this is recombinant DNA, so you have mice that are now expressing a gene for bioinflorescence that they don't normally have. We can get mice like that by genetic engineering. And so this is going to be the anytime you manipulate genes for practical purposes. Not that making glowing mice is very practical. A more practical example would be using E. coli to produce human insulin. So now you're using a bacteria to make insulin, which is necessary in humans. And so when you talk about diabetes and needing insulin for diabetics, E. coli can be used to produce that. So biotechnology is the use of living organisms or their components to perform practical tasks. So again, another example would be using bacteria to digest oil that would make it into the environment through an oil spill. One pretty common thing you're going to see a lot, and we're actually going to work with restriction enzymes in a lab. Restriction enzymes are used to cut DNA at very specific nucleotide sequences. We call those areas restriction sites. And once that DNA is cut, you can actually insert new genes into that strand of DNA and then splice it back together. So these restriction enzymes are going to be obtained from bacteria. As I said, we will be working with some restriction enzymes for a lab in class. So what we have here is our double-stranded DNA. We have our restriction enzyme recognition sequence. So this is where the restriction enzyme is going to go. It's going to cut the DNA. And notice that it's a jagged cut. It doesn't cut it straight through. And that's really important. These overhanging ends are called sticky ends. They're important because when you add a new sequence of DNA in here, like this new gene, the sticky ends are going to match up. So here you had TTAA left over. You can see here you have an overhanging end on the new gene, which is AATT. And so they will complementary base pair, and now you have a new gene inserted into the original DNA sequence. And so this is one new combination. DNA ligase would then seal the strands, and you've now used a restriction enzyme to cut open a strand of DNA and insert a new gene. Plasmids are something else that are used just extensively in biotechnology. Again, we'll be talking about plasmids for a lab that we're going to do, and these are essentially a vehicle for transporting genes. We will discuss very briefly other vehicles that are used to insert genes, but plasmids are going to be the big one. So when we use plasmids, there's 4 specific steps that we have to use. First, you have to get the DNA for the trait that you're looking for. Once you have that DNA, then you need to get it into the plasmid. Then you go through a process called bacterial transformation, again, a lab we're going to do. And then you try to identify the new trait. You look to see if the DNA for the trait made it into the bacteria. So here we have our plasmid. All right, we have our restriction site, and so again, use a restriction enzyme to open that up. Here we have the human DNA that has the gene that we're trying to add to the plasmid. So you use the same restriction enzyme to cut both so that they have matching sticky ends. You are going to then mix the DNAs. They're going to join by base pairing. Then you're going to add DNA ligase, and what that's going to do is it's going to cause the base pairs to bond. So now you have a human DNA fragment that is in a plasmid. Next, you're going to put the plasmids into bacteria through bacterial transformation. And then you're going to let the cells divide, and then you are going to end up with lots of bacterial cells that contain the plasmid with the gene of interest. And so now you can try to identify whether or not this was successful and whether or not the gene of interest has made it into the bacteria. So with insertion, you're actually placing the foreign DNA into the plasmids. You open the plasmid with the enzymes to create the sticky ends, splice the new DNA and plasmid together. Transformation then is placing the plasmid into the bacterial cell. Again, very important lab, and you're going to need to know it for the AP test. In order to do this, we're going to use temperature shock and salt treatment. You could also use electric current. We will use a temperature shock. And then identification is simply screening the altered cells to see if the desired gene is there. We will be doing this with antibiotic sensitivity. You could also look for a new trait. So maybe the gene was for a new color, or in the case of the mice, it was to make something glow. So those are all ways to identify if the gene made it into the bacteria. So some ways that this could be used, we talked about E. coli making insulin, human growth hormone, or just a way to make proteins that we need for like pharmaceuticals or things like that. So here we have essentially the same thing being described, but down here we have some different applications for using these plasmids. So here we have a gene that's being inserted into plants. This one is showing that a gene could be used to alter bacteria so they can be used to clean up waste. This could be a protein that dissolves blood clots in heart attack therapy, or then we also have that human growth hormone, and it can treat stunted growth. So genes can't be above a certain size in order for this to work, and you also can't need to splice the gene to remove introns in order for it to work. So there are some limitations. It can't be too big, you can't have introns, so this isn't going to work for everything or for every gene, but it will work for certain ones. So DNA sources. You can get DNA from an organism by using a section of their chromosome. You can use complementary DNA or cDNA. This is a created copy of DNA from the mRNA transcript, and what that does is it avoids introns. So if you needed a gene and you wanted to use the DNA of that gene, if there were introns, then that gene is no good for using bacterial transformation. However, if you got an mRNA transcript of that gene after it had been processed, you now have that complete mRNA transcript already processed, all the introns removed. So you can then use reverse transcriptase, and you can go from the completed mRNA transcript back to DNA that we call the complementary DNA, and now the introns are gone because they've already been removed from the mRNA. And so that is one way to use a gene that has introns. It's to use reverse transcriptase on the mRNA transcript. Again, this is just showing you how you use reverse transcriptase. So here is the gene of interest. You can see it's got introns. So if you get the mRNA transcript, then you can remove the mRNA transcript. So here we're in a test tube, use reverse transcriptase, and now you're making this complementary strand of DNA with no introns. And so now this is a gene that you can actually use where before you couldn't. So what other ways can we do this? So what if we have bigger genes that we want to get into a plasmid? Well, we can't. So we have to have other vehicles to do this. And so you have bacterial artificial chromosomes that can handle much larger inserts. So now we're looking at 100 to 300 kilobase pairs. You have yeast artificial chromosomes, which are even larger. And then you also have human artificial chromosomes. All right, next we have nucleic acid probes. These are going to be used to find a specific DNA sequence in a mixture of DNA pieces. So DNA is going to be denatured and then cut up to produce pieces. A piece of complementary DNA is added as a probe, and the probe has been labeled. And then you're going to look for where the probe goes in the DNA sample, and that's essentially going to lead you straight to sequences of DNA that you're looking for. The next thing we're going to talk about is PCR, or polymerase chain reaction. So think about a chain reaction, like you have one event that sort of sets off this series of events, and that's kind of how you think of PCR. So you start with one strand of DNA and then it leads to lots of strands of DNA. So this is a method for making copies of a specific segment of DNA in order to amplify it. So you start with one copy, you end up with lots of copies, you are amplifying that DNA. So how do we do PCR? You are going to separate the strands by heating, which would denature the DNA. Then you're going to cool it down slightly, and then you'll build the new strand from primers and nucleotides, and then you just keep repeating that sequence until you have as many copies of the DNA as you need. So here we have the targeted sequence of DNA. You're going to heat it briefly, and when it's heated up, it's going to separate, and then you're going to cool it down. That will allow primers to hydrogen bond. DNA polymerase will then copy the nucleotides the same way as replication always happens. And so you started with the one strand. You have two strands, you keep repeating this, and by cycle 3 you now have 8 strands of the DNA that you're looking for. So it can amplify DNA with as little as 1 original copy. This is very useful in a variety of different tests. So again, if you're really into like the NCIS, I think that's what it's called, or CIS, those kind of crime shows where they talk about, getting DNA samples. Sometimes DNA samples are really small, you don't have a lot of DNA to work with, so you can use PCR to amplify that sample. Next we have gel electrophoresis. This is a technique that will be covered in the lab. It's used to separate mixtures of DNA or proteins. And it uses restriction fragment length polymorphisms. So it's a method for detecting minor differences in DNA structure between individuals. Again, if you're into those crime shows, it's very common in DNA fingerprinting. So what you first do is you're going to digest that DNA. You're going to break it up into smaller pieces using restriction enzymes. You will then separate the DNA by gel electrophoresis and then identify sequences with probes. And so the results will give you patterns of DNA markers, or what we call a DNA fingerprint. And then the markers are inherited in Mendelian pattern, and so they can be used to show relationships. So sometimes when you're watching some of these crime shows, they'll say, oh, like this isn't the guy, but we know he's related to our suspect. And this is one of the ways that they can kind of see that. And so here you're looking at just a classic completed gel. You'll a lot of times see pictures of this on TV shows. And so what happens is you've got these different bands of DNA segments that are moving a certain distance. The distance is controlled by how large the fragments are. So larger DNA fragments are not going to move as far in a gel as shorter DNA fragments. So it's kind of like anything else. Think about like a really small person walking through a crowded hallway versus a really large person. That small person is going to be able to kind of move through that area a little bit faster, kind of dodge people. Whereas a larger person is going to have trouble moving through a crowded hallway. Same kind of thing. Longer fragments won't move as far. Shorter fragments will move much farther. So it's a way of breaking up the different DNA fragments and kind of comparing them. DNA sequencing uses dideoxynucleotides, which stop DNA polymerase at a specific point in DNA replication, and it will build new DNA from a single strand of DNA, and it will produce fragments of different lengths. So this is important because it's read base by base, so you can look at overlapping pieces of chromosomes and essentially get an entire genome of an organism that can be read. Next we have cloning. This is reproducing organisms by asexual means, very common in plants. A lot of plant cells show totipotency, which means that you can take one small piece of a plant and you can end up growing a new plant from it. Cloning in animals is a little bit different. It's done by nuclear transplantation. The main example is Dolly, the sheep that was cloned in the mid-90s, so started in 96. I believe she was born in 97. Lots of other things have now been cloned. And what happens is you're taking mammary cells, you're taking the nucleus from one cell and a mammary cell from another cell, bringing them together. You'll get an early embryo. That early embryo is then implanted into another surrogate mother, and then you get your lamb. So stem cells are unspecialized cells that can reproduce itself or differentiate into other cells. There are embryonic and adult stem cells. Embryonic have been in the media a lot. They're used a lot for research, so can we use them to replace damaged cells or damaged body parts? You'll hear a lot about this in the media. I encourage you guys to look it up. And I don't want to give away too much because your lecture question is, why do you think so many types of DNA technology are controversial, and do you agree with this? So I know this was a long, fast lecture, but please send me a message with DNA technology and controversy. All right, thanks guys.